also need to subscribe my channel and give a like to my live stream. Okay. Cell unit of introduction. All the living organisms are composed of cells. Cells are of two types, unicellular organisms and multicellular organisms. So unicellular organism, single cell, multicellular means the many cell body cell. Cell is the functional structure, fundamental unit of a living organism. Unicellular organisms are capable of independent existence, performing the essential functions of a life. Anything less than a complete structure of a cell does not ensure independent living. Living cell first seen and described by Anton von Leeuwenhoek, and nucleus was discovered by Robert Boyle. The inventions of the microscope and this improvement leading to the electron microscope. Electron microscope improvement revealed all the structural details of the cells. Cell theory is given by Matthias Seligman and Theodor Swan. Examined by large number of plants, observe that all plants are composed of different kind of cells. These cells from the tissue of this plant. So, Matthias Seligman, German botanist in 1838, examined a large number of plants. Observed that all plants are composed of different kind of cells. These cells form the tissue of the plant. Theodor Swan, British zoologist, 1839, studied different types of animal cells. He reported that the cell had a thin outer layer. Theodor Swan reported that the cell had a thin outer layer, which today now known as the plasma membrane. Based on his studies on plant tissues, Swan said or concluded that cell wall is the unique features of our plant cell only. Swan proposed the hypothesis that bodies of plants and animals are composed of cells and product of cells. Thus, Seligman and Swan formulated the cell theory. Cell theory did not explain how cells were formed. Exception of cell theory. RBC and sieve tube do not have nucleus and other organs, but still they are considered as a living cell. Virus lacks cellular machinery. Rudolf Virchow. In 1855, he modified the hypothesis of Seligman and Swan. He given the cell theory a final shape. So he first explains how cells were formed. Rudolf Virchow first explains that cell divided and new cells are formed from pre-existing cells. Cell theory as understood today, all living cells are composed of cells and product of cells. All cells arise from the pre-existing cells. Viruses are considered as an exception to the cell theory since they lack the cellular machinery. And the sieve tube and RBC do not have the nucleus and other organelles, but still they are considered as a living cell. This is also an exception. Now, the overview of the cell. It's a onion cell is given in NCERT. It is a typical plant cell. It has distinct cell wall as its outer boundary. Just within the cell wall is the cell membrane. Cell wall, it is the outer boundary of the typical plant. It's typical cell. Example, onion cell. Cell membrane. This membrane lies inner to the cell. It acts as a delimiting structure in case of animal cell. Example, human chick cell. Nucleus. Inside its cell is a dense membrane-bound structure called nucleus. The nucleus contains chromosome, which in turn contains the genetic material. So, eukaryotic cells and prokaryotic cells having nuclear membrane-bound nuclei, and prokaryotic cells like membrane-bound nuclei. Cytoplasm, present in both prokaryotes and eukaryotes, it is a semi-fluid matrix. It occupies the volume of the cell. The cytoplasm is the main arena of the cellular activities in both plants and animals. Various chemical reactions occur in it to keep the cell living. Cell organelle. The eukaryotic cells have other membrane-bound distinct structures called organelle. Examples of organelle are Golgi complex, lysosome, ear, vacuoles, microbodies, mitochondria. Prokaryotes lack such organelle. Non-membrane-bound organelle example is ribosome. Ribosome present in both prokaryotic and eukaryotic organisms. Ribosomes are found in cytoplasm, chloroplast, mitochondria, and trachea. Inclusion bodies are also non-membrane-bound organelle. Animal cell contain another non-membrane-bound organelle, that is centrosome, which help in cell division. Size of a cell. Mycoplasma, it is the smallest cell and its length is 0.3 micrometer. 1 micrometer equal to 10 to the minus 6 meter. Bacteria's length is 3 to 5 micrometer. Ostrich egg is the largest and the single egg. Human red cell length is 7 micrometer in diameter. Nerve cell is the longest cell. It is branched and long. Shape of the cell may vary with the functions they perform. They may be polygonal, cuboidal, columnar, thread-like, and irregular, and even irregular. So name and their size. Mycoplasma, its size is 0.3 micrometer. Bacteria 3 to 5 micrometer. RBC's diameter is 7 micrometer. Typical bacteria is 1 to 2 micrometer. PPLO, that is 0.1 micrometer. Viruses 0.02 to 0.2 micrometer. Eukaryotic cell is 10 to 20 micrometer. Prokaryotic eukaryotic cell. Cisternae Cisternae is the part of Golgi complex and crystin is the part of mitochondria. Cisternae diameter is 0.5 micrometer to 1 micrometer. Mitochondria is 0.2 to 1 micrometer. Ribosome is 15 nm to 20 nm. Ribosome is 15 to 20 nm and cisternae is 0.5 micrometer to 1 micrometer. Mitochondria, that is 0.2 to 1 micrometer. Prokaryotic cell. Prokaryotic cells are represented by blue-green algae, bacteria, mycoplasma, pleuropneumonia-like organism. Prokaryotic cells are generally smaller and multiply more rapidly than the eukaryotic cell. Prokaryotic cells are generally smaller. Prokaryotic cells are generally smaller and multiply more rapidly than the eukaryotic cells. These may vary in shape and size. There are four basic shapes of bacteria: spherical shape, rod shape, comma shape, and spiral shape. Spherical shape, that is the coccus single. Rod shape, that is bacillus. Comma shape, vibrio. Spiral shape, spirillum. Spherical shape, coccus. Rod shape, bacillus. Comma shape, vibrio. Spiral shape, spirillum. The organizations of prokaryotes is functionally similar, even though the prokaryotes exhibit a wide variety of shape and size. All prokaryotes exhibit a cell wall surrounding the cell membrane except mycoplasma. Mycoplasma has no cell wall. Prokaryotes have no well-defined nucleus. The genetic materials of prokaryotic is basically naked, not enveloped by nuclear membrane. What is plasmid? Is the genomic DNA, single chromosome, circular DNA. Many bacteria have small circular DNA outside the genome. So, what is plasmid? Is genomic DNA, single chromosome, circular DNA. Many bacteria have small circular DNA outside the genomic DNA. Small circular DNA is the plasmid. The plasmid DNA confers the unique phenotypic characters to such bacteria. One such character is the resistance to antibiotics. Plasmid DNA is used to monitor bacterial transformations within the prokaryotes. Now the nuclear membrane is found in prokaryotes. No organelle like the one in eukaryotes found in prokaryotes except ribosome. Prokaryotes have something unique in the form of inclusion body. Prokaryotes have something special in the form of inclusion bodies. What is mesosome? It is the characteristics of prokaryotes, a specialized different form of cell membrane called the mesosome. Mesosome are essentially the enfoldings of cell membrane. It is a special membranous structure. It is formed by the extensions of plasma membrane into the cells. The extensions are in the form of vesicles, tubes, and lamina. What are the function of mesosome? They help in cell wall formation, it helps in DNA replication, it helps in distribution to daughter cells. They help in respiration and secretion process. These help in increase the surface of the plasma membrane and the enzymatic content. So mesosome increase the surface area of the plasma membrane and enzymatic content. They help in cell wall formation, they help in DNA replication, they help in distributions of daughter cells. They help in respiration and secretion process. They help to increase the surface area of plasma membranes and enzymatic content. What is chromatophore? In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called the chromatophore. They contain pigments, cell envelopes of the prokaryotes. Most of the prokaryotes, particularly the bacterial cell, have the chemical complex that is known as cell envelope. Cell envelope consists of tightly bound three layer, that is glycocalyx, cell wall, plasma membrane. Glycocalyx is the outer membrane. Glycocalyx differ in composition and thickness among the different bacteria. If it could be loose sheath, that is the slime mold. It could be thick and tough, that is the capsule. It is a middle layer. Cell wall is the middle layer. The cell wall determines the shape of the cell. It provides strong structural support to the bacteria to prevent the bacterium from the bursting or collapsing. Cell membrane, it is the inner layer. It is selectively permeable. It interacts with the outside world. The membrane is similar structure to the eukaryotes. The bacteria are classified into two groups on the basis of different in the cell envelope, the manner they respond to the staining procedure developed by the gram. So gram positive, when they respond to gram, that is gram stain, that is the gram positive, and when they do not take the gram stain, that is the gram negative. Movement of bacteria. Bacteria cells may be motile or non-motile. Bacteria have pili and fimbriae as the structure. Flagella, pili and fimbriae. Flagella help in motility. These are the thin filamentous extensions that arise from the bacterial cell wall. Bacteria show a range in the number and arrangement of the filament. It has three-part filament. Longest portion arising from the cell wall structure to the outside, hook and basal. Pili are the elongated tubular structure. It is made up of special protein, that is pillin protein. These are the smaller pistol-like structures shooting out of the cell. In some bacteria, it helps bacteria to attach with the rock in stream. It helps in attachment of the host cells. The fimbriae help in attachment of the Yes, these are the small bristle-like fibers sprouting out of the cells. In some bacteria, it helps bacteria to attach the rock. In skin, it helps in attachment to the host tissue. Pili is made up of pilin proteins. Flagella is a thin membranous. Pili is elongated and fimbriae is small bristle-like structure. Ribosome in prokaryotes, ribosome are associated with the plasma membrane of the cell. Prokaryotes contain 70S, 50S plus 30S. Eukaryotes contain 80S, 60S plus 40S. Size is 15 nm to 20 nm. As a site of protein synthesis, sometimes ribosome may attach to a single amino and form the chain that is the polyribosome or polysome. The ribosomes of the polysome carry out the translations of amino into protein. These materials in prokaryotes are stored in the cytoplasm in the form of inclusion bodies. These are the non-membrane bound, lie freely in the cytoplasm. Inclusion bodies, example is phosphate granules, cyanophane granules and glycogen granules. And the gas vacuoles are found in blue-green algae, blue-green bacteria, purple bacteria and green photosynthetic bacteria. Eukaryotic cells. Eukaryotes include protists, plants, animals and fungi. They have the membrane bound organelle. Eukaryotic cell possesses an organization of the nucleus with nuclear envelope. There is an extensive compartmentation of cytoplasm through the presence of membrane bound organelle. Eukaryotic cells have variety of complex locomotor cytoskeleton structure. Their genetic material is organized into chromosome. All eukaryotic cells are non-identical. Plants and animals are different as the former possess several plastic central vacuoles but absent in animals. Now the vacuoles is a large central vacuoles in plant and it is other smaller in animals. Cell membrane plasma membrane is found inside the cell after invention of electron microscope. The detailed structure of cell membrane plasma membrane was studied. Electron microscope help in chemical study of cell membrane, study of RBC reduce the possible structure of plasma membrane. Main components of plasma membrane is lipid, mainly phospholipid, phosphoglyceride, second protein and carbohydrate cholesterol. Major lipid are phospholipid and they are arranged in bilayer. Lipids are arranged within the membrane, head towards outside and tail inside. This ensures the non-polar tail are saturated. In humans, membrane of liquid protein is 52%, lipid 40%. The phosphate or the phosphatic membranes are cholesterol. Integral proteins are partially or totally buried inside the cell. Peripheral protein lies on the surface of the cell. An improved model of the structures of cell membrane was proposed by Singer and Nicholson. Fluid mosaic model discovered by Singer and Nicholson. The quasi-fluid nature of lipid enabled lateral movement of protein within the overall bilayer. This ability to move within the cell membrane is measured is predicted. The Singer fluid mosaic model discovered by Singer and Nicholson in 1972. The quasi-fluid nature of lipid enabled lateral movement of protein within the overall bilayer. The quasi-fluid nature of protein enables the lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of membrane helping function, that is of cell membrane fluid nature. The fluid nature of membrane helping cell growth, formation of intercellular junctions, secretion, endocytosis and cell division. So the fluid nature of membrane helps in cell growth, formation of intercellular junctions, secretion, endocytosis, cell division. Cell growth, formation of intercellular junctions, secretion. Function of membrane. Main function is transport of molecules. The membrane is relatively permeable to some molecules present on either side of it. Membrane is selectively permeable to some molecules present on either side of it. Active transport and passive transport is that movement is a movement of molecules or ions in across the membrane against the concentration air, low concentration, high concentration energy will be released in A plus and K plus. It is the movement of molecules across the membrane along the concentration between high concentration to low concentration without expenditure of energy. There are two types. Passive transport, simple diffusion, it is the movement of neural solids across the membrane. Osmosis, it is the movement of water by diffusion across the membrane. Polar molecules cannot pass through the non-polar lipid as they require the carrier proteins. It is the non-rigid cell wall. It is a non-rigid living structure. Cell wall found outside of the plasma membrane of fungi and plant. Cell wall are two types: primary cell wall, secondary cell wall. Primary cell wall is a young plant cell which by primary cell wall growth and gradually diminishes as the cell matures. Secondary cell wall is formed on the inner side towards the inner membrane side of the cell. Middle lamella is made up of calcium pectate. It helps or glues the different neighboring cell together. Cell wall and the middle lamella may be traversed by plasmodesmata which connect the cell cytoplasm of the neighboring cell. Algal cell wall made up of cellulose, galactans, mannans, and minerals. Plant cell wall is made up of cellulose, hemicellulose, pectins, and proteins. Function of cell wall. Cell wall not only gives shape to the cell, it also protects the cell from mechanical damage. It also helps in cell-to-cell interaction. It provides barrier to the undesirable macromolecules. Endoplasmic reticulum is endomembrane system is endoplasmic reticulum, Golgi complex, lysosome vacuoles. In electron microscopic structure of eukaryotic cell reveals the presence of network of reticular tiny tubular structures scattered in the cytoplasm. ER divides the intercellular space into two compartments. ER divide. Endoplasmic reticulum divides the intercellular compartment luminal and extraluminal. Luminal and extraluminal. Endoplasmic reticulum has luminal and extraluminal. After electron microscope discovery, reveals the presence of network of reticular tiny tubular structure that scattered in the cytoplasm. So the ER often show the ribosome attached to the outer surface. The RER, that is rough endoplasmic reticulum ribosome on the surface called the rough endoplasmic reticulum, and absence of ribosome appears smooth. Rough endoplasmic reticulum is frequently observed in the cells actively involved in protein synthesis and secretion. ACR major site for synthesis of lipid. In animal cell, lipid-like steroid hormones are synthesized by ACR. They extend to outer membranes of the nucleus. RER present in the eukaryotic cell, ACR found in the eukaryotic cell. ER contains tubules and cisternae. ER helps in transport of substance, synthesis of proteins, synthesis of lipoproteins and glycogen. Synthesis of protein. Golgi complex flat disc shape, cisternae 0.5 to 1.0 means micrometer in diameter. Discovered by Camillo Golgi, densely strained reticular structure near the nucleus. They are stacked parallel to each other. Wide number of cisternae present in the Golgi complex. The Golgi complex are concentrically arranged near the nucleus within the distinct convex seas. The forming phase, concave trans, the mature phase. Convex seas. The forming phase. Convex is the forming phase. Concave, the mature phase. The seas and trans phase of the RNA are entirely different but interconnected. Function of Golgi apparatus help in packaging materials and to be delivered either to the intercellular targets or secreted outside the cell. Golgi complex connected with the endoplasmic reticulum because the material to be packed in the form of vesicles from the endoplasmic reticulum fused with the seas phase of the Golgi complex and moved towards the concave trans. A number of protein syntheses by ribosome on the ER are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi complex is important site of formation of glycoproteins and glycolipids. Is the question of NEET 2019 and 2020. Lysosome. These are the membrane-bound vesicle structures formed by the process of packaging of the Golgi complex. Lysosome is a single membrane-bound or structure. Isolated lysosome vesicles have been found to be very rich almost all type of hydrolytic enzymes. Hydrolysis, lipase, protease, carbohydrates. They optimally activate acidic phase. Ribosomal enzyme digest carbohydrate, protein, and nucleic acid. Vacuoles. These are the membrane-bound surface found in the cytoplasm. Vacuole contain water-soluble excretory products and other materials not useful for the cell. Vacuole is bounded by single membrane called tonoplast. In plant cell, the vacuoles occupy only 90% of the volume of the cell. In plant, the tonoplast facilitates the transport of ions and other materials in the concentration gradients in the vacuoles. Hence, their concentration is higher in vacuoles than the cytoplasm. In amoeba, the contractile vacuole help in osmoregulation or excretion in many cells like protists. Food vacuoles are formed by engulfing of the food particles. Singular form of mitochondria is mitochondria. Mitochondria clearly visible when their strength, number, shape and size of mitochondrial particles are variable depending upon the physiological activity. Shape of the mitochondria shortest shaped or cylindrical having diameter 0.2 to 1 micrometer, average 0.5 micrometer, length 1.0 to 4.1 micrometer. Mitochondria double membrane structure, outer membrane and inner membrane. It has two compartments, outer and inner. Inner is filled with dense homogeneous substance, that is the matrix. Inner and outer membrane is dividing its lumen distinct into two aqueous compartments. Inner compartment and outer compartment. Outer compartment it is smooth. It is formed by continuous remaining boundaries of the RNA. Inner compartment. Inner membranes form a membrane component string. So matrix possess circular DNA, few RNA, ribosome component for protein synthesis. True membranes have its own specific enzyme associated with their mitochondrial function. Aerobic respiration, it produces solar energy in form of ATP, so is the powerhouse of the cell. Mitochondria help in oxidative phosphorylations and generations of adenosine triphosphate. Mitochondria divides by fission.
one chapter I've revised in biology.